Hi everyone, welcome to Siva's Science Galaxy. I am planning to give series of lectures on protein structure, diversity and folding. I trust the information will be very useful to the students and research scholars studying protein chemistry. All the videos will be kept under biology category in playlist of the channel. In this video lecture, I'll be talking about structural architectures of proteins. First, let us discuss about what are proteins. Proteins are biological macromolecules and the proteins are essential components of cells, tissues and organs of our system. Hence, we can say proteins are building blocks of life. Interestingly, proteins provide about one third of one's daily calorie intake. In this context, it is curious to know the foods that are rich sources of proteins. Food materials such as milk, meats, grains, eggs, nuts, and seeds are rich sources of proteins. Now let us discuss about biosynthesis of proteins. In our system, proteins are synthesized in the ribosome of human being. The proteins that are synthesized in the human being are made up of amino acids that are linked by peptide bonds. Now we have two questions. What are amino acids? What do you mean by peptide bond? Now let us discuss about amino acids. Amino acids are chemical molecules having both amino group and carboxyl group in their structures. Depending upon the position of amino group and carboxyl group, the amino acids can be classified into many types, such as alpha amino acids, beta amino acids, gamma amino acids, and so on. In this slide, I am showing the model structure of alpha amino acids. In the alpha amino acid, the amino group and carboxyl group are attached to a carbon atom. The chemical moieties consisting of amino group, carboxyl group, and proton can be called as backbone of the amino acid. The R group can be called as side chain. Depending upon the R group, there are many types of alpha amino acids. In other words, in the alpha amino acids, the backbone is similar in all the cases. The amino acids are differing from one another in their side chain. Now, in the previous slide, we answered to the question, what are proteins? Proteins are made up of amino acids that are linked by peptide bond. Now, we redefine the definition of proteins. What are proteins? Proteins are built by alpha amino acids that are linked by peptide bonds. As I mentioned in the previous slide, proteins are synthesized in the ribosome of human being, and the proteins that are synthesized in the ribosome are exclusively made up of alpha amino acids. More interestingly, the proteins that are synthesized in the ribosome are exclusively made up of 20 standard alpha amino acids. The 20 standard alpha amino acids can be called as proteinogenic amino acids. The proteinogenic amino acids can be classified into many types on the basis of their chemical properties. As you can see in the slide, the proteinogenic amino acids have been classified into non-polar amino acids and polar amino acids. Amino acids such as alanine, proline, valine, leucine are good example for non-polar amino acids. Similarly, amino acids such as serine, threonine, asparagine are example for polar amino acids. Depending upon the nature of side chain, the proteinogenic amino acid can also be classified into aromatic amino acids, positively charged amino acids, and negatively charged amino acids. Tyrosine and tryptophan are the good example for aromatic amino acids. Amino acids such as lysine, arginine, histidine are positively charged amino acids because they have positive charged chemical moiety in their side chain. Amino acids such as aspartic acid and glutamic acid are example for negatively charged amino acids. I have taken these informations from the Fundamentals of Biochemistry by White and White. For further information on the topic, you may refer the book or you may refer the book you wish to read. Now, as I mentioned the previous slide, the proteins that are synthesized in the ribosome are exclusively made up of alpha amino acids. There are two types of alpha amino acids. Depending upon the position of the chemical moieties in the three-dimensional space, the alpha amino acids can be classified into alpha L amino acids and alpha D amino acids. Interestingly, proteins that are synthesized in the ribosome of human being are exclusively made up of alpha L amino acids. So now, what is the answer to the question, what are proteins? Proteins are built by alpha L amino acids that are linked by peptide bonds. Now, I trust that we have a clear understanding on amino acids. Now let us discuss about peptide bonds. What are peptide bonds? Peptide bond is a bond formed between a carboxyl amino group of one amino acid and amino group of another amino acid. In other words, the backbone carboxyl group of one amino acid and backbone amino group of another amino acid combine and give a peptide bond by eliminating a water molecule, as you can see in the slide. So, two amino acids combine and give a peptide bond. Three amino acids combine and give a two peptide bond. So, a polypeptide chain consisting of n amino acid will give n-1 peptide bond. I hope that now we are very clear about what are amino acids and what is peptide bond. Now, we have a very clear answer to the question, what are proteins? Proteins are made up of alpha L amino acids that are linked by peptide bonds. Now let us discuss about various structural levels of proteins. There are four structural levels: primary structures, secondary structures, tertiary structures, and quaternary structures. What is primary structure? The amino acid sequence of protein can be called as primary structure. In other words, the linear polypeptide chain is known as primary structure of proteins. So what is secondary structure? Secondary structure is a structure built by backbone interactions of the amino acids present in proteins. What is tertiary structure? Tertiary structure is a structure built by side chain interactions of amino acids present in proteins. What is quaternary structure? Quaternary structure is structures among the polypeptide chains in multimeric proteins. Multimeric proteins are the proteins having two or more than two polypeptide chains. So quaternary structure is a structural interactions between the two polypeptide chains in a dimeric protein, or the structural interaction among the polypeptide chain in a multimeric proteins. Now let us discuss about three-dimensional structures of proteins. The three-dimensional structures of the proteins can be elucidated by two techniques that are available to date. The two techniques are NMR, nuclear magnetic resonance spectroscopy, and X-ray crystallographic methods. These are the only two techniques available to date to determine the structures of the proteins at high resolutions, in other words, at atomic level resolutions. In this slide, I am showing the three-dimensional structure of myoglobin and hemoglobin. The myoglobin and hemoglobin are the two proteins for which three-dimensional structures of the proteins could be determined by using X-ray crystallographic techniques. The myoglobin structure was determined by John Kendrew and his team. The hemoglobin structure was determined by Max Perutz and his team. John Kendrew and Max Perutz received Nobel Prize for their contribution towards the structure determination of the proteins myoglobin and hemoglobin. They received the Nobel Prize in chemistry in the year of 1962. As I mentioned, NMR is another technique by which the three-dimensional structures of the protein can be determined at high resolution. In this slide, I am showing the 3D structure of the protein called BBTA. BBTA stands for bovine pancreatic trypsin inhibitor. The structure was elucidated by Kruth-Utterich and his team. Kruth-Utterich received Nobel Prize for his monumental contribution towards the NMR of proteins in chemistry in the year of 2002. So as I mentioned in the two previous slides, NMR and X-ray are the only two techniques by which the structure of the protein can be determined at high resolution by experimental methods. The three-dimensional structures of the protein can also be predicted by three popular bioinformatic tools. The three popular bioinformatic tools are homology modeling, threading, and ab initio. Homology modeling and threading are template-based method. Ab initio is a template-free method. In the case of homology modeling, we have a target structure and template structure. Target is the protein for which sequence is available, structure is not available. Templates are the protein for which sequences as well as structures are available. Using the three-dimensional structure of the template, the structure of the target can be predicted. Similarly, in the case of threading, based on the three-dimensional structure of the template, the target structure can be determined. The main difference between the homology modeling and threading is, in the homology modeling, the structures of the target and template will be very similar. In the case of threading, the three-dimensional fold of the target and template will be very similar. As I mentioned, ab initio is a template-free method. The method works on the basis of the first principle. In other words, the three-dimensional structures of the protein can be predicted using the first principles of the method. I hope the informations are very useful for you. If you have any clarifications, suggestions, or questions, please comment. Thank you very much for your attention.